All right, today we are diving into a biological mystery that honestly sounds like it's straight out of science fiction. We're talking about a creature that can literally cheat death by drying out completely, and the incredible, sophisticated molecular toolkit that makes it all possible. So let's just jump right in. This is a survival story, but it's not your average one. It's about a tiny creature that has totally mastered an unbelievable survival strategy. I mean, it sounds impossible, doesn't it? If you lose all the water in your body, well, you're basically dust. But, in the natural world, it seems like for almost every rule, there's a fascinating exception. And here's our exception. This little guy is a tiny, non-biting midge from Africa, and its larvae can survive being almost completely desiccated, basically dried out. They can wait for years in this, seemingly lifeless state until the next rain comes along and just brings them back to life. So, how in the world does it pull off this incredible feat? Well, the answer has a fancy scientific name, and it's a process that has absolutely fascinated biologists for decades. Anhydrobiosis. It's a kind of cryptobiosis, which means hidden life. The organism isn't dead, not at all. It's just paused. All of its metabolic processes just grind to a halt, waiting for the right conditions to fire back up again. So here's the midge's brilliant three-step survival plan. First, it completely floods its cells with a special sugar called trail hollows, which basically takes the place of water. Step 2, this sugary mix turns the inside of the cell into a solid, glass-like state, a process called vitrification, which locks everything in place so it doesn't break. But even that's not enough to prevent all damage. So, for the crucial third step, it deploys a specialized army of protectors. And these are the cell's molecular bodyguards, LEA proteins. They are unbelievably good at stopping other important molecules from clumping together and falling apart as the cell dries out. They are absolutely essential for surviving this whole process. But here's where things get interesting. When scientists started looking at the midge's genome, they found a real puzzle. It wasn't just, a few of these protective proteins, it was a whole arsenal. 27. The sleeping chironomids has genes for 27 times different kinds of these LEA proteins. For one tiny organism, that is a staggering number. And that became the central mystery. Are they all just doing the same job? Are they redundant backups, maybe? Or is something way more sophisticated going on here? This is exactly what a team of researchers decided to figure out. Okay, so to solve this puzzle, they had to see these proteins in action. They needed a way to follow each and every one of those 27 guards on its patrol inside a living cell. Their method was actually really clever. They took the gene for each of the 27 proteins and fused it with a gene for green fluorescent protein. You might know it as GFP. This basically sticks a glowing green light on each protein. So when they put them back into cells, they could literally watch under a microscope and see exactly where they went. Okay, so here's their very first clue. This chart shows which proteins are being made and when. As the midge starts to dry out, you can see some proteins pop up in huge amounts right away. But others? They're made at different times or in different amounts. This staggered deployment was the first big hint that these proteins aren't just copies of each other. They probably have different jobs to do at different stages of the process. And then the mystery got even deeper. So on the left, you're seeing one of these proteins inside the midge's own cells. See how it stays in the main part of the cell, but it's kept out of the nucleus, the control center? Now look at the right. The scientists took that exact same protein and put it into a different type of insect cell, and its behavior totally changed. It just went everywhere, including the nucleus. This was a huge deal. It showed that a protein's location isn't fixed. It actually changes based on the cell it's in. And the evidence for specialization just kept piling up. Look at this image. This protein isn't just floating around randomly. No way. It is specifically stationed at two key locations, the endoplasmic reticulum and the Golgi apparatus. You can think of these as the cell's crucial factories and shipping centers. So this isn't some random bodyguard. This is elite security assigned to protect the most vital infrastructure in the cell. This table really sums up just how weird and inconsistent the protein's behaviors were. So in the midge's own cells, a protein like PVLEA3 has a very specific job, guarding the cell's factories. But stick it in another cell, it just kind of wanders around. Another protein is blocked from the nucleus in one cell, but in another, it's allowed right in. The takeaway here was crystal clear. This is not a simple system of one protein, one job. It is all about context. So what does all this complexity actually mean? And more importantly, what does it mean for us, for potentially harnessing this incredible ability one day?
The researchers themselves say it perfectly in their paper. This inconsistency, it's a critical piece of the puzzle. It means you can't just take one of these proteins, drop it into, say, a human cell, and just expect it to work the same way. The cellular environment itself is part of the instruction manual.
So the answer to that whole 27 protein puzzle is that the complexity is the answer. This isn't just a toolbox where every hammer does the same thing. No, it's a highly coordinated team of specialists. Each one has a specific role, a specific location, a specific time to act, and all of that can change depending on the cell they happen to be in. So let's just recap what we've learned from all this. Surviving being completely dried out isn't about one single magic bullet protein. It's a huge coordinated effort. These LEA proteins are highly specialized, and what they do is deeply connected to the cellular environment they're in. And what that means is, if we ever want to borrow this superpower, we can't just focus on one star player. We have to understand the entire team. And this brings us to a final, pretty mind-bending thought. The damage that freezing causes is a massive hurdle in medicine, especially for things like organ transplants. But what if, by understanding this entire system, from this tiny little midge, we could learn to put human tissues, or maybe even whole organs, into a state of dry, suspended animation, at room temperature, just waiting, ready to be revived whenever they're needed. It's still a long way off for sure, but the secrets to doing it might just be hidden inside the midge that cheats death.